Okay, so welcome back. This is going to be our very first screencast for Chapter 12, and in Chapter 12 we are going to be looking at sponges. And so if you look at the very top of this screen, you're going to notice that we have three different varieties of sponges. And one thing I want you to really pay attention to is the complexity or variety of shape that you're going to find in each of these animals. Some sponges are going to be rather tube-shaped. Some of them are going to be, well, still in tubes, but they're going to be a little bit thicker. And then, of course, this third one is going to be more of a branched type of sponge. Now, when it comes down to the evolution of sponges, a lot of scientists have theorized that sponges must have originated from what we call aggregates of cells. And many scientists believe that those aggregates were most likely unicellular protozoans, and those would be the organisms that we looked at back in Chapter 11. So we're going to look at sponges, and we're going to look at different characteristics, we're going to look at behaviors, and we're also going to look at varieties of sponges that you might find in nature. So looking at this first slide, we need to look at a little bit of the evolutionary history of sponges. Now, sponges are thought to have originated from a special type of single-celled organism called a choanoflagellate, and that's what you're going to see represented right here. Now, these choanoflagellates can be solitary or they can be colonial, which means they can be by themselves or they can exist in groups. Now, each cell has a flagellum, and you can see the flagellum right here, and it's surrounded by a collar of microvilli, which are used to collect tiny particles, and most often these particles are going to be little bits of food. So the body of the cell is located right here, and right here you're going to notice this is the collar. And all of this right here, in other words, this kind of net-like appearance that you see surrounding the flagellum, that's going to be the microvilli. And as we said before, they're going to act as a very tiny and very efficient type of filter. Now, many of these organisms are sessile, which means that even though they do have a flagellum, they do tend to stay put in one place. Now, these are important because they tend to resemble the feeding cells that are found in many sponges that are out there. Now, we call these feeding cells choanocytes, and that's where we got the name. So they're very similar to the choanoflagellates. Now remember, these are examples of an actual single-celled unicellular organism. Choanocyte, like the one you see down here in the lower right, this is actually a cell that was taken from a sponge. And so what I want you to do is I want you to notice the similarities between the two. If you notice, we definitely have the flagellum, which is located right here in the middle of the cell. You can see the flagellum here, and we also have the collar, which is located right here, and you can see sort of the sieve-like appearance of the microvilli. And they do the same thing that they did in the choanoflagellates. They work to filter the water. And as they filter the water, they're taking out very tiny food particles. And then, of course, we have the body of the cell right here. And then the only difference between these two is that the choanoflagellate does not have this amoeba site connected to this cell. This amoeba site is going to be there to basically take the food that has been brought in by the microvilli. That food is going to be passed down the body of the cell, and then it's going to be taken up by the amoeba site, which is basically going to function as the last step in digestion for these animals. And so the big thing for this slide is to see the similarities between the choanoflagellates, again this single-celled protozoan, and the choanocyte, which is found inside lots of sponges that we're going to look at. So the phylum that we're going to be looking at is the Phylum Porifera. Now, porifera means pore-bearing. So what we have is we have an organism that is made up of lots and lots and lots of very tiny holes. And so we call these tiny holes pores. Now, these holes are important because what they do is they act to draw water into the body of the animal. Now, as they draw the water in, they're also going to draw food in. And so by using the choanocytes that we had discussed in the previous slide, that's going to be the way that they actually feed. So remember, these choanocytes were these flagellated collar cells. And so again, we have a cell like this with a flagellum, and then we have that collar that surrounds the flagellum. And those microvilli are going to act to filter the water and basically trap the food. Now again, these choanocytes not only are going to trap food, but they're also going to act to remove waste as well. Now most sponges or most members of this phylum are going to be marine, but there's actually about 150 freshwater sponges out there, and there's even a few that are considered brackish water. Now brackish is going to be like a combination between fresh and salt water. They tend to range from about a few millimeters to almost 2 meters in length. So there's a big difference in size with the sponges. Now even though sponges tend to be sessile, they tend to be stationary when it comes down to most of their life, the young are considered free swimming. So when these animals reproduce and they produce young, they're going to be free swimming for a short period of time, and that's the way they're going to distribute themselves throughout their environment. So when talking about the phylum Porifera, it's really important that we make sure to talk about the different types of relationships that can be formed between sponges and other animals. There were 3 different types of relationships that we talked about back in Chapter 11 when we discussed protozoans. We had talked about a mutualistic type of relationship, and this is where one member would benefit and actually the second member would benefit as well. We talked about a parasitic type of relationship, and this is where one member would benefit and actually the other would be harmed in some way. And then we talked about this one right here called a commensalistic type of relationship, and this is one where one member would benefit but the other member is not helped, but it's not harmed either. Now there are various different animals that will form these relationships with sponges. Some examples would be crabs, nudibranchs, which are like sea slugs. Some mites will form relationships with sponges, and there's even some fish that will live in sponges. And again, these are going to be commensal, which means that this shrimp over here is going to benefit in some way. Maybe it's protection, maybe it's in its ability to maybe take food from the sponge. But the sponge itself really isn't harmed in this situation. Now when you talk about the phylum Porifera, we talk about members of this phylum that, believe it or not, actually do have sort of a rudimentary type of skeleton. Now their skeleton can be made either of a fibrous type of material or a more rigid type of material. Now if it's a rigid type of material, it's going to be made of either a calcareous material, which is a material made of calcium carbonate, or a silicous type of material, which is going to be made of silica. Now when you talk about these two components, you're talking about something that is called a spicule. So the spicules are very rigid, and if you look over here on the right hand side, you can see how rigid these spicules can be. They come in lots of different shapes, lots of different sizes. In fact, you can find some that sort of look like stars, there are some that look like snowflakes, there's even some that look like rods. Now what's really interesting about the spicules is that some scientists can actually determine what type of sponge it is based on the shape of spicules that you will see. Now the fibrous portion of some sponges is going to be made of something called collagen, and collagen is going to make up the fibrous portion called spongin. So over here on the right hand side you're going to see an example of spongin fibers. Now again, this is more of a flexible type of material. It's more fibrous in nature, and it doesn't have the organization that you would find in spicules, so it's a bit more random in how they would be put together. So when you think about a sponge, you often think about something that can be squeezed or maybe can retain water like a typical kitchen sponge would. Well, if you're looking at a sponge that is able to do that, that's able to actually hold on to maybe water, for example, then you're probably talking about a sponge that is made primarily of spongin. Now there are some of them out there like the glass sponges, which are primarily made of spicules, you know, this silica or calcium carbonate type of material. And those would be ones where if you tried to squeeze them, they're probably going to fall apart because these are very rigid structures. So you need to be aware of both of these. You need to be aware of spicules and you need to be aware of spongin and the difference between the two. Now when you mention a sponge, most people think to themselves a sponge is something that can hold on to water. Now when it comes down to sponges that you find in nature, that's still the case, depending on the type of material that they're made out of. Now sponges have something called an internal canal system, and the reason it's called an internal canal system is that we have water flowing continuously from the outside to the inside of the sponge. Now the water is going to enter the sponge through a special structure called a dermal ostia, and these are going to be the small openings that you're going to find in the pinacoderm, which is kind of like the skin of the sponge. Now water is going to be directed past the choanocytes. Now back at the beginning of our screencast we had mentioned that the choanocytes were really important when it came down to collecting food for the sponge. And they're going to collect this food as the water passes by using those microvilli. Now remember, we have a choanocyte that has a cell body, and you can see that right here, and we have a flagellum, which extends out of one end of the cell. And then, of course, we have the collar of microvilli. And so as the water moves past those microvilli, it's going to trap the food. It's going to bring the food down through the body of the cell, and eventually at this end of the cell is going to be an amoebocyte, and that amoebocyte is going to finish up digestion of that food for the sponge. Now, there also might be some accessory cells like pinacocytes and archaeocytes, which also might participate and help in the feeding of the sponge. So when you look at sponges, what you're going to do is you're going to group those sponges into three main body designs. We have one body design that's called the ascon or the asconoid type of body design. We have one called the cycon or cyconoid type of design, and then we have one called the leucon or the leuconoid body design. Now when you look at the ascon, you're going to notice that it's a pretty simple sponge. You have a large interior space of the sponge that's going to be lined actually with choanocytes, and these choanocytes are going to be located right here. And if you remember back in our previous slide, we had talked about this being the pinacoderm of the sponge, or sort of like the skin of the sponge. And you can see the openings or the dermal ostia located right here. But what makes this one different from the two that you see off to the right hand side is that there's a very linear arrangement to the way that this sponge is put together. We still have a very large opening at the very top where the water is going to come in through the ostia, and it's going to leave through something called the osculum at the top of the sponge. So this one is put together pretty simply. Now when you talk about a cycon or a cyconoid type of sponge, you still have basically the same body shape. You do still have an area inside of the sponge that is pretty significant. It's pretty large. But in this case, when you look at the pinacoderm of the sponge, you're going to notice that it's been folded in many times. Now, you still have choanocytes that are going to line the folds that you see in this sponge, and they do exactly the same thing. They're still going to function in helping that sponge to collect food. You still have dermal ostia, but those ostia are going to be found maybe here, 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 and here. And you still have a large major opening at the very top, which is still going to be called the osculum. So really the only difference between these two is the infolding that you see of the body wall of the sponge. Now when you look at the leuconoids, now this is where things get a little bit different. First off, you'll notice that the osculum at the very top of the sponge has been greatly reduced. So the size here is definitely much smaller than you would see here. Now if you look at the ostia, you're going to notice the ostia are definitely increased. So you have lots of different areas where that water can enter the sponge. And you're also going to notice that we have some larger canals through here. So that main interior canal that we saw right here has now been broken into several smaller canals. And these are called radial canals. Now if you notice, we still have choanocytes in the sponge as well because they're important because they help the sponge in regards to feeding, but these choanocytes are going to be located and grouped in small circles within the sponge. And if you notice, water is going to collect in these areas, and so the filtering of the water to collect food is still going to take place. Now that water is going to pass out through here, and it's actually still going to make its way back up, and it's still going to pass out of the sponge through the osculum. Now one of the big differences also with this sponge is that first off, this arrangement is going to increase the surface area for this sponge, which means there's more opportunities for feeding within the sponge. And what that's going to do to benefit the sponge is it's actually going to allow these sponges to get much larger. So the more surface area you have, the larger the sponge can become. So these tend to stay small, not a lot of surface area, a little bit more infolding, going to give a little bit more surface area, so these tend to get a little bit larger, and then the leuconoids can actually be the ones that maybe get up to about 2 meters in length. Now something that's also kind of interesting about the leuconoids is that because the opening up here is so incredibly small, when they do release water from the sponge through this opening, that water can be forcefully ejected from the sponge. So oftentimes if you take a leuconoid out of the water, they will forcefully eject the water out of the sponge. So they can eject this water maybe 2, 3, 4, maybe 5 feet. So it's a pretty powerful stream of water that you're going to find leaving the sponge just because of the size of the opening. Alright, so that's going to wrap up this first screencast for Chapter 12. Please make sure that your study guide is completed before you come to class.